Good morning, students. So today we are going to discussing the diagram come activity based questions, the 10th class biology, chapter 6th, reproduction and the generating system. So already you know that reproduction definition, the ability of an organism to give rise to new generation of individuals or offsprings or descendants is called reproduction. Reproduction are done mainly, it replaces the new one, important one. And let us go to the 33 diagram based come activity question. So here, what does this diagram is indicate? This diagram is indicate that there will be a reproduction method, asexual reproduction method. The organism name is paramecium. Paramecium undergo binary fission. Not only paramecium, but also euglena, bacteria also undergo fission. Yes, daily life bacteria undergo binary fission. Explain the process of organism that is undergoing. These organisms undergo the binary fission, longitudinally binary fission, and one organism split into two. The process of dividing one organism into two equal, it is called fission. Because of two organisms, it is called binary fission. So because of it is a unicellular organism, paramecium, amoeba, euglena, bacteria, majority they are the unicellular, so that they oftenly undergo asexual reproduction method, that is particularly binary fission. Binary means two, fission means division. So one parent organism undergo division, produce two daughter cells, equal cells. These are becoming as a parents. So one organism is dividing into two, that is called binary fission asexual reproduction method. This binary fission or asexual reproduction method mainly which organism follow means unicellular organism. Unicellular organisms example paramecium, euglena, amoeba, bacteria. So next to 34. What does this diagram is indicate? This diagram is indicate that yeast cells. These are the yeast cells. Name the organism found in this picture. So number of yeast cells we can see this. So these are yeast Saccharomyces cerevisiae, and because of there will be a innumerable of yeast cells we are seeing from the one parent cell. So this is the one parent cell. On the one parent cell, developing number of birds like structures, these separated and form a new organism. So number of birds are forming. Because of forming on the surface of the parent yeast cell, number of birds, it is called budding. So budding is the reproductive method, asexual reproduction method, budding. This budding process oftenly followed by the yeast. Yeast belongs to fungi. Yeast organism, it is a unicellular organism. It belongs to fungi. Oftenly reproduced by the budding. Budding is one of the asexual reproduction. Budding is one of the asexual reproduction. Next 35. What is the organism present in this diagram? So this diagram, what is the organism present in this diagram is Spirogyra. What is the type of asexual mode of reproduction found in this diagram? Fragmentation. 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 Explain the process of seen in this diagram. So the detached fragment of this one separated and developed into a new organism, that is called fragmentation. A piece or a small part of the parent organism is separated and developed into a new organism is called fragmentation. And give some more examples about fragmentation. We can take about bacteria, algae, some of the members. Examples. So what does this diagram is indicate? This diagram is indicate that regeneration is the one of the reproductive method. What is the organism found in this planaria? Could it be known as a type of fragmentation? Why? Yes. Why? Because when we remove head part and tail part, head is developed a tail part, tail is developed body and head part. So that here what's happened? They're gaining the lost part. Whatever they lost, again they're developing their parts. So it is called regeneration as well as fragmentation. Why? Because a part of the organism is separating and developing into a new organism. It is nothing but a fragmentation. One of the asexual reproduction method. The process is mainly planaria. Example, which process means regeneration. Regeneration. Next, what type of vegetative propagation is seen in this diagram? And so what type of vegetative propagation is seen in this diagram is cutting. So here what happens, a part of the plant, twig, we are separating and sowing into a pot, and then it developing a new plant. So a part of the plant we are separating and sowing into the pot and developing a new plant is called cutting. We are separating a vegetative part. Vegetative part is nothing but a root, stem, leaves. So give some examples for plants that are propagating this marigold. We can take hibiscus example, hibiscus and marigold, rose. These are the plants we can propagate with the help of a process is called cutting. Explain process briefly. So the part of the, before taking a part, we can select a nodal region where the leaf is there. In that region we have to cut. Why? Because from the nodal region, when we separate and sow into the soil, that part will develop the roots, whereas after the node, if we separate, that part is not develop the roots. So that always when you are taking the part of the plant, you must see the nodal part, because nodal part where it is, we can see where the leaf is developing. In that region below the leaf developing, that part you have to take, because of that is the nodal region. leaf developing part and the stem is nothing but nodal part. That part is have ability to develop the roots. So from that part you have to detach from the plant and sow into the soil, develop into a new plant. That is called cutting. Example, hibiscus roses sinensis, rose plant and marigold plant. We can easily possible to show the cutting. And next 38, what does this diagram is indicate? And this diagram is indicated by the mainly layering. Example, Nerium and Jasmine. So what does this diagram is indicate? This is the vegetative propagation method, layering. One of the vegetative propagation method is layering. So layering is followed in by which plants means Nerium and Jasmine. The plant which is developing the branches near the soil and bent down and keep a wooden block formed to attach the soil. After a few days, roots are developed and we have to cut here and the detached now when it is separated and develop into a new plant. So this is called layering. Because of we are using the underground near the ground part, it is called ground layering. It is called ground layering. And air layering also is there. That is another process. And now let us grafting. Grafting. So how many plants are involved in this process? Two plants are involving. What is the plant which is attached to the soil or called? So whatever we should call that is what so now this part is called root stock and this is called scion or graft. So the graft part does not have root system. The root stock part have the roots. So now we are this part is cut taking from the another plant. So now this is called scion or graft. Now the root part plant is called root stock. What is the other plant without roots? Without a root plant is graft or scion. Without a root, it is called graft or scion. And the root part is root stock. Name this process of vegetative propagation is grafting. Give example for this type of propagation is mainly follow the mango, citrus, apple. Mango, citrus, apple. So grafting అనేది ఈ plants లో possible అవుతుంది. Mango, citrus, apple. So by this once again I will conclude that here the 37 is cutting, 38 is layering, 39 is grafting. So cutting, layering, grafting, these are the modern vegetative propagation methods. Today we are using more frequently to develop innumerable of plants and having good quality within short period in limited time and limited area. We are using by the process vegetative propagation. So now the 40, what does this diagram is indicate that? It is a tissue culture. What are the plant parts can be used for this purpose? Mainly, either we take in tissue culture a leaf portion or tissue or cell, whatever may be. Cells part we take, nothing will happen. Organ part, leaf completely we take, nothing will happen. So, shall we produce parthenocarpic plants in this method? Yes, we can produce parthenocarpic plants without the fusion of gametes directly from the male and female organs by using vegetative parts. By using vegetative Name the organisms seen in this diagram. So Rhizopus, it is the fungal organism. To what division of organism does it belong? Fungi, fungi, and it is bread mold. It is also called bread mold. What are the black ball-like structures are called sporangia. Inside spores are present. It is a sporangiophore. The stalk-like structure is called, these are the root-like structures are called rhizoids, and these branches are fungal hyphae. Branches are called fungal hyphae. The branches like structures are called sporangiophore, which develop at the terminal part is sporangia. Inside spores are present. Each spore is responsible to develop a new fungi. So sporangia. Explain the process of asexual reproduction seen in the diagram. So mainly sporangiospore or sporangia by which asexual reproduction is occurred. So these are the bread mold or rhizopus belongs to fungi. And next 42. Name the diagram and label the parts. So it is the human reproductive system. When you label the parts, so it is a main. So when you take these are the testes. These are the testes. The inside the folded part is called epididymis, and this is called penis, and the urethra, prostate glands, seminal vesicles, ejaculatory duct, and as well as vas differentia. Vas differentia. So these are the parts. In which part of this system produces sperms? Mainly testes, seminiferous tubules. టెస్టిస్ లోపల సెమినిఫెరస్ ట్యూబుల్స్ అని ఉంటాయి. ఆ సెమినిఫెరస్ ట్యూబుల్లో జెర్మినల్ ఎపిథీలియం అనే టిష్యూ ఉంటుంది. ఆ జెర్మినల్ ఎపిథీలియం టిష్యూ నుంచి స్పెర్మ్స్ ప్రొడ్యూస్ అవుతాయి. విచ్ హార్మోన్ is responsible for this means testosterone. What are the two glands seen in the system means seminal vesicles and prostate glands and Cowper's glands. Prostate glands and Cowper glands we can see in this. So now name the diagram and label the parts. So this is the human sperm diagram. So the parts is, it consists mainly four major parts: tail part, middle piece, neck, head. Head have a sharp point, it is called acrosome. Inside there will be a nucleus is present, and the middle piece, it consists neck region, there will be a mitochondria, and how it is structure suitable for its function. So the structure is responsible to carry out a mainly it is a male gamete. And why does it contain a portion of head, number of mitochondria, and a long tail? So long tail is responsible for to swim in the semen, and the middle piece consists mitochondria for energy production, and the head region have acrosome to penetrate into the ovum. Next to 44. Name the diagram and write the different parts of the picture. This diagram is indicate that human being female reproductive system. The main parts is there will be a vagina, cervix, and uterus, a pair of ovaries, and a pair of fallopian tubule and one uterus. These are the parts of female reproductive system. And what are the ovule produced in this system? Where are they? In the abdomen near region, ovule are produced inside the ovary. So here inside the ovaries, ovules are produced. These are the ovaries. There will be two ovaries are there from which ova are produced. Write the process of ovulation. The process of releasing of ova by the ovary is called ovulation. What are the hormones released in the system? Progesterone, estrogen, and these are responsible to production of secondary sexual characteristics in the female reproductive system. And the next 45, write the stages of the fetus in this diagram and explain the three layers that help in the fetus. So mainly, so three stages of the fetus is amnion, chorion, and here umbilical cord is present, and there will be amnion and chorion. So amnion is helps to the by the amniotic fluid. This fluid is helps to mechanical shocks and jerks. So chorion is surrounding, which is the tissue is belongs to between mother and fetus, and there will be umbilical cord which is connected to the abdomen of the fetus, supplies the nutrients from mother to fetus. And 46, name the diagram and label the parts. So this is you can see easily, this is the datura flower, datura metal, and what we can say, this datura in Telugu umeta puvu. So the pods is mainly, it is the sexual organ in plants. So flower, it is the sexual organ, sexual reproductive organ in the plant. The pods is mainly, this is called pedicel. The green colored outer part is called calyx. Yellow colored part layer is called corolla. And these are the stamens. And there will be a centrally located part is called, this is the stigma, style, and ovary. Inside the ovary, ovules are present. So this is the sexual reproductive organ. So name the diagram and label the parts. It is a datura flower. It is the sex organ in the plant. What are the essential and non-essential parts of the plant? So the flower is generally consists of four floral parts, and two are essential, two are non-essential. So two are essential are called third and fourth whorl, androecium and gynoecium. Stamen and pistil are the essential organs, sex organs, whereas calyx, corolla, non-essential organs which are present outside. So the flower has four floral parts, and in which outer two are non-essential, lower two are essential. So what are the two diagrams indicate? These two diagrams indicate that it is the pollen grain, and pollen grain is developing in the pollen tube. This pollen tube, and how many nuclei are present in the second diagram? What are they? So in second diagram, what happens? The pollen grain developing the pollen tube. This is the pollen tube, which consists two male nuclei. First daughter male nuclei, second daughter male nuclei. How is the tube formed and entered into the ovule? This tube is formed from the pollen grain, cell wall, and protrude and form a pollen tube and penetrate into the ovule. Pollen grain is the male gamete, which is formed in the stamens, particularly anther, inside the anther. So name three of them and write the. So here loop of Henle and vasectomy and tubectomy. These are the preventive methods, that is to control the pregnancy. These are the three methods. One is the loop of Henle, vasectomy, and the tubectomy. These are the methods to prevent the pregnancy. Three instruments. Purpose of these is to control the birth control methods. So these three instruments already explained you. Once again, I'm explaining. First one is the loop, the vasectomy and the tubectomy. These are the methods used in the birth control methods. And 49, name the diagram and label it. The diagram is indicate for the fourth floral part. Flower lo unde center lo unde 20 last part. So it is gynosium. The gynosium has three important parts. The pot-like structure is called ovary. The stalk-like is called style, and the upper one is called stigma. So ovary, style, stigma. Ovary, style, and stigma. And inside the ovary, there will be a ovule is there. These ovules have a female gametophyte. And how many ovules are there in this diagram? Only one ovule is there. And how many nuclei are there in this embryo sac? Total 8 nuclei are present, and 3 are antipodals, 2 are synods, 1 egg cell, and 1 is the center cell. And how does double fertilization occurs in the diagram? So double fertilization is occurred. First fertilization is occurs between egg cell and first male daughter nuclei, and second fertilization is occurred another male daughter nuclei with the secondary nucleus. So because of two male nuclei in the pollen grain and fused with the two, one is the egg cell, another one is the central nuclei. Because of occurring two fertilizations, it is called double fertilization and triple fission, which is a unique feature in the plants. And next, what does this diagram is indicate? Name the different phases. Explain the different phases of this cycle and what happens if M phase is missed in this M cycle. This cycle is indicate that mainly mitosis, and that is cell division. It is cell division, cell cycle. It is called cell cycle. The cell cycle is consist total 24 hours. Cell cycle. This cycle is consist of mainly two phases, M phase and interphase. సో సెల్ డివిజన్ లో సెల్ సైకిల్ ఉంటుంది. సెల్ సైకిల్ అంటే బర్త్, గ్రోత్, డెత్. అంటే డెత్ అయ్యేటప్పుడు 2 cells నిచ్చిపోతుంది. టోటల్ గా దీన్ని సెల్ సైకిల్ అంటాం. సెల్ సైకిల్ కన్సిస్ట్ 2 important phases. One is the mitosis and second one is the interphase. So the interphase is consist, so already I said the total cell cycle to occur 24 hours. The interphase has 23 hours and mitosis have only 1 hour. In 23 hours, what's happened? 3 important processes occurred: G1, G2, and S phase. In S phase, what happened? Synthesis phase and gap 1 and gap 2. These 3 should take the 23 hours. M phase means one cell to gives rise to 2 cells is only in M phase. In mitosis, it takes only 1 hour. So in 1 hour, from one cell, 2 cells are developing, which are exactly. So this is called cell division and cell cycle. So these are the important diagram come activity based questions in the reproductive lesson. So you once again you go through, take a textbook and go through these question answers and practice and write down